Trees grow in many distinct ecosystems, from the harsh cold climate of the boreal to the humid sauna of the tropics. These conditions permit growth rates that differ greatly. For trees that grow in areas with distinct seasons, like temperate zones, predictable periods of more rapid and less rapid growth alternate as spring and early summer turn to fall and winter. Such alternating periods of growth do not occur in the tropics. Looking at these two temperate trees, what can we say about their ages? Is the gargantuan tree on the left much older than the little tree on the right? As it turns out, from the outside of the tree, we actually have few clues about the ages of these trees. But if we look inside the tree, we see a different story. Here is what is colloquially termed a cookie, or a cross-section of the tree's trunk. Here we see the tree's pith, or center, the heartwood, which is actually dead tissue, the sapwood, in which the cells are still alive, the vascular cambium, which continues to divide and increase the diameter of the trunk, and lastly the bark, or the outermost protective cortex of the tree. The sapwood and heartwood, which make up the majority of the tree's volume, are actually composed of xylem tissue, which the trees use to conduct water and soluble mineral nutrients from the soil and the roots up towards the leaves. By counting the number of dark rings, which represent the slower growth of the tree during fall and winter, we can get an accurate idea of how old the tree was when we cored it or when it died. Think back to our two trees from before. Believe it or not, such analyses of tree rings have shown some Krumholtz trees to be hundreds or thousands of years old, which is as old or even older than this gigantic tree on the left. We can learn other things about the tree we want to study, and in a much less invasive way than cutting out a full cookie from the tree's trunk. By using an increment borer, also called a tree corer, we can take a thin core out of the trunk and leave the rest to keep growing, which any anthropomorphized tree would appreciate. Once we have taken cores from the tree we want to study, we can look at other characteristics of the xylem in these cores. For example, we can look at how thick different rings are and also perform more meticulous studies of the xylem's cellular structure to infer how growing conditions may have changed throughout our tree's lifetime. Individual trees can grow for hundreds or thousands of years, but scientists have managed to extend how far back in time dendrochronology can bring us, so to speak, by studying the annual ring patterns of trees which grew in different, though overlapping, time periods. By aligning periods at the start of one tree's lifetime with the end of another, older tree, researchers can establish a dendrochronology that spans these two trees' lifetimes. Adding more trees to these data has enabled the compilation of thousands of years of tree growth, giving us one more source of data with which to model how climate has changed in our past. What limitations are inherent to dendrochronology? Well, although you can infer how a tree's growth rate has changed, it can be hard to know which factors cause these changes. For example, a period of poor growth could be caused by a lack of nutrients, or erosion of the soil, the presence of pests or diseases, or even increasing neighborhood interactions such as competition with neighbors as the canopy closes. For our class project, we studied how plant communities and climate have changed at the Daly Point Nature Reserve, a protected park near Bathurst Harbour in New Brunswick. This site is a salt marsh, which is an ecosystem that can form between the land and the sea. Salt marshes are built up by sediment brought in by the tides and deposited there, especially around the roots of salt-loving halophytic grasses. But it can also be destroyed by strong storms and anthropogenic disturbances. Specialized plants like Spartina alterniflora and patens thrive here because of their adaptations to such conditions. For example, they use parenchyma, which help oxygenate their roots and the soil just around these roots. The rest of our study group focused on the abundances of different kinds of pollen that have been preserved in distinct layers in the mud of the salt marsh, while I looked at how we can use dendrochronology to gain further insight into how this site has changed over time. We took 3 cores from the biggest red spruce tree that we found on a mound of relatively dry dirt in the salt marsh. We sanded and mounted the cores, as was described in the instructive videos of James Dyer, and then counted the tree rings. We also grouped periods into years of good, moderate, and poor growth, based on the size of the pale rings of growth during spring and early summer. I then compared climatic data from the Bathurst Airport Station to these periods to see if there were noticeable changes in temperature or precipitation regimes between the periods in which the tree was growing quickly and when it was growing poorly. Statistical tests that I ran in R showed me that precipitation was the most likely factor affecting tree growth, among the factors that I assessed. Precipitation may interact with air pollution to produce acid rain, which would decrease the availability of soil mineral nutrients like calcium. This could reduce the winter hardiness or resistance of red spruce trees. How sure am I of this result? And how transferable is this hypothesis to the rest of the marsh? Unfortunately, because of the very small sample size of one tree, and because of the difference between trees and grasses, and relatively dry soil in salt marsh mudflats, it is difficult to claim that this result may be quite relevant for Daly Point Nature Reserve at large. Future work could expand on the number of trees that were cored to allow a more thorough comparison of how climate may be changing tree and salt marsh plant growth. Thank you so much for watching this short video prepared by me, Chris Madsen, for my class project in Biology 550 at McGill University.